In this lesson, we will be covering Chapter 10.8 through 10.10 and 10.15 through 10.18. We're first going to look at some evolutionary trees. These trees help us to conceptualize and categorize biodiversity. Evolutionary trees can also be called phylogenetic trees. A phylogeny is the evolutionary history of organisms. So these phylogenetic trees are going to be one way that we can look at the evolutionary history of organisms and which other groups are closely related to them. We can see that in some cases our classification system matches very well with these phylogenetic trees. In this case we have order Archaea, which has to do with the number of toes these animals have. There are two families here, Bovidae and Cervidae. And then 4 genuses, so the tree branches 4 times for each genus, and then 4 species that correspond with the 4 animals at the top. I want to point out, though, that this is not always the case. Sometimes our traditional classification system does not reflect the evolutionary history of the animals. And in these cases, we're often recategorizing, regrouping, and reorganizing our classification system. Let's look at some of the basics of how to read an evolutionary tree, or a phylogenetic tree. We're going to start with the nodes, or branch points. These nodes show us the point of divergence, where 2 groups branched and started to become different. This also shows us the site of a common ancestor. On this tree, this node here is the site of the common ancestor for mice, rats, humans, birds, and fish. Trees have many branch points or nodes along them. Here we see another branch point. This is the common ancestor of mice, rats, humans, and birds, but not fish. Fish branched earlier and are not included in this lineage. We can see another common ancestor here, this one for mice, rats, and humans. And again, it does not include birds because birds branched earlier and are behind this common ancestor. And the last common ancestor on this tree is between mice and rats. These 2 groups are closely related, therefore close on the evolutionary tree. Because rats and mice are so closely related and they have a very recent common ancestor, we say that these groups are sister taxa. It's important to note that when we look at these nodes or branch points, these evolutionary trees can be rotated along those branch points without actually changing anything in the tree. So we see that this tree over here, the mice and the rats are still sister taxa. Even though this tree has been rotated on its node, this is the same tree because the relationships are the same. These trees can have species at the end of the branches, or they can have major taxa or groups from our classification system. We can look here again and find examples of sister taxa. So the badgers, Taxidea taxus, and the otters, Lutra lutra, are sister groups. And then the Canis latrans, the coyotes, and the Canis lupus, the wolves, are also sister taxa. And remember, our nodes are the site of common ancestors, so every time we see a branch point, we know that there's not only a common ancestor, but this is a point of divergence where the 2 groups have become different. This tree is another example of how our traditional classification system, so order, family, genus, species, lines up very well with an evolutionary tree. So order Carnivora is one branch. We see that Felidae branched first, okay, it's a family. The mustelids and the Canidae, so dogs, weasels, and their ancestors, branched separately. And there are 4 genuses with the 5 different species at the ends. Here's a practice question. Which animals are more closely related according to this phylogenetic tree? Take a minute to figure this out. The correct answer is crocodiles and birds. These taxa are closely related. They're sister taxa because they have a recent common ancestor and they are located right next to each other on this phylogenetic tree. The overall goal of phylogenetics is to determine evolutionary relationships and place all living organisms into monophyletic groups. A monophyletic group is a group in which all individuals are closely related to each other, and they're more closely related to each other than to any other outside group. This means that when we place these organisms onto a phylogenetic tree, a monophyletic group will include a common ancestor and all the descendants branching off of it. Here's an example. We can see that birds and crocodiles are a monophyletic grouping. We can include common ancestor A, birds and crocs, and have everything that branches off of it. So this is monophyletic. If we want to put together the group reptiles, crocodiles and lizards, this is not a monophyletic group. We have common ancestors A and B included, and some of the descendants, but not all of them. Because the birds are left out, this is not a monophyletic group. When constructing evolutionary trees, we use comparisons of traits and DNA to help us determine the relatedness of the organisms. In this example, we see different traits, and we look at whether these organisms have them or do not have them. So in this case, the retractable claws are seen in both lions and hyenas, but not in bears and wolves. The number of ear bones in lions and hyenas are 2, but only 1 in the bears and the wolves. And the more similarities these animals have, the more closely related they are. So we see in this example, African lions and spotted hyenas are sister taxa, which are more closely related to each other than they are to the other groups. Black bears and gray wolves are sister taxa, more closely related to each other than they are to the other groups. We can also do DNA-based evolutionary trees where we compare differences in DNA base pairings to determine relatedness of these organisms. Again, when we compare these organisms, we see that there are more similarities between species 1 and species 2 than there are to the others. This makes species 1 and species 2 more closely related, so on this tree they are sister taxa. The most accurate phylogenetic trees tend to use the most types of data they can find, both physical features and DNA sequences. The more evidence you have to back up your phylogenetic tree, the more strongly supported your hypothesis is. We have to keep in mind, though, that similar structures don't always reveal common ancestry. We have looked at analogous structures before, traits that look similar but have come from different evolutionary lineages and have arisen because of convergent evolution. We see that wings in bats and wings in insects are analogous. They are both meant for flight, but they evolved independently. We see many analogous structures. The shape of a seal, a dolphin, a shark, and a penguin are all hydrodynamic. These animals are all torpedo-shaped. However, these animals are not closely related. 2 are mammals, one's a fish, and one's a bird. This means that this trait has arisen through convergent evolution because they all live in a similar aquatic habitat. Remember, these analogous structures have arisen due to convergent evolution. They are the opposite of homologous features, or homologous traits. These are features that have been inherited from a common ancestor. These traits may look slightly different or have variations on a theme, but they are similar due to common ancestry. A good example of this are the bones found in the forelimbs of tetrapods. All 4-legged animals have the same bones in their arms, even though they are modified in different ways for different lifestyles. So how do we figure out if a trait is homologous or analogous? One way is to observe multiple traits in an organism to determine relatedness. Another way is through DNA analysis. We can also find the opposite to be true, that some animals and some organisms that do not look very similar may be closely related. A good example of this is the African golden mole and an African elephant. The mole and the elephant are more similar to each other based on their DNA sequence than the golden mole is to the common shrew, even though the common shrew and the golden mole may look more similar using their physical features. DNA sequences often reveal closely related organisms and hidden evolutionary histories that we were previously unaware of. Many scientists are working to put all living organisms together into one giant tree of life. We are not going to delve deeply into the diversity of life, but we are going to investigate the three domains of life: domain bacteria, domain archaea, and domain eukarya. All living organisms on Earth are divided into one of these three categories or domains. Bacteria and archaea are both prokaryotic. Domain eukarya is named after the eukaryotic cell. We can see that domain archaea and domain eukarya are sister taxa. So remember, we've looked at the two cell types in Unit 1. So this is a review question for you guys to work on. Think of all the similarities and differences between prokaryotic and eukaryotic cells. If you don't remember, you may want to take a few minutes and pause this video to go back and review the similarities and differences between these cell types. Bacteria and archaea are both prokaryotes. These are tiny cells with no nucleus and no organelles. All of these organisms are single-celled. Domain bacteria includes the vast majority of prokaryotic species familiar to most people. These organisms tend to live in more neutral environments than those found in domain archaea. Bacteria have one or more circular chromosomes of DNA, and they always reproduce asexually, making clones of themselves using a process called binary fission. Bacteria are found all around us in the world. They are in the soil, in our environment, on the surfaces we touch every day. They are on our bodies and in our bodies. We cannot escape them no matter what we do. Domain archaea is also prokaryotic. It also has one or more circular chromosomes of DNA. These organisms also reproduce asexually. However, most archaea are extremophiles. This means that they love to live in extreme places. They can do this because of the unique linkages they have in their membranes. This allows them to survive very harsh environments. Some examples are archaea that live in hydrothermal vents in the Pacific Ocean. Here we see that temperatures can rise as high as 121 degrees Celsius. These archaea are adapted to this condition and thrive in these areas. We see them deep sea as well in the hydrothermal vents at the bottoms of the ocean. We see that these archaea, this yellow sludge on the sides of these rocks, survive very well, and they use the weird combination of compounds and chemicals coming out of these vents as an energy source. Archaea are very diverse and are often divided into 5 groups based on their features and what areas they tolerate. So thermophiles tend to love to live in hot places. Halophiles are highly tolerant of salty regions. There are some that are very tolerant of high and low pH levels, some that are tolerant of high pressures, and then there are the methanogens, which are a weird group that produce the compound methane. Methanogens are often found inside of other organisms, such as in the intestinal tract of cows and other ruminants. The third domain is domain eukarya. These organisms all have eukaryotic cells, which means they have a nucleus with many chromosomes that are linear and organelles to help get jobs done. Some organisms in the domain eukarya are single-celled, while others are multicellular. Some can reproduce asexually, but most also have sexual reproductive cycles. Domain eukarya is highly diverse. It includes protists, which are often single-celled organisms. Protists also include the algae. We see plants, animals, fungi, all found in this group as well. Domain eukarya is broken down into 4 kingdoms: Kingdom Protista, Kingdom Plantae, Kingdom Fungi, and Kingdom Animalia. Again, diversity here is huge and ranges from large multicellular organisms like whales and giant trees, all the way down to single-celled organisms such as Paramecium or Stentor, these little protists seen here. In summary, we have looked at phylogenetic trees and the domains of life. Be sure to look at the learning objectives found in Blackboard to help you review this lesson.